Hello everyone, welcome to another tutorial from Biology with Shalini, and in this video I'm going to talk about collagen types and synthesis and some of the medical conditions associated with it. Now we know that collagen is the major structural protein in extracellular matrix. Collagen is made up of amino acids, and as we know, amino acids are made up of carbon, hydrogen, oxygen, nitrogen. So it contains specific amino acids like glycine, proline, hydroxyproline, and arginine. Like glycine, proline, hydroxyproline, and arginine. So it is single most abundant protein in human body, constituting more than 25%, approximately 30% of all proteins. And it is present almost everywhere in our body. It is found in bones, tendons and ligaments, and of course skin, heart, kidney. So this collagen, it is important in suppleness and constant renewal of skin cells. It is the cement that holds everything together. Collagen and keratin, they are fibrous proteins and insoluble in nature. So important point will be fibrous protein. As we know that myoglobin and hemoglobin, they are globular proteins. Insoluble, right? Insoluble in water. It is long, inelastic, triple-stranded helical structure, and we have to remember that the helix is not alpha helix, right? So fundamental unit of collagen is tropocollagen, and it is a triple-stranded helical structure. These alpha chains will wind around each other and they will form a triple-stranded helical structure, and 3.3 amino acid residue per turn will be there, and they wind around one another in a characteristic right-handed triple helix. Okay, so the main point is fibrous protein, insoluble in water, helix is not alpha helix, and this helix is stabilized with the help of hydrogen bonds. Hydrogen bond help them to wind around each other. And this hydrogen bonding is not between the amino acids, it is between the chains. Amino acid sequence in alpha chain is glycine XY, where X can be any amino acid, but mostly it is proline, and Y will be 3 or 4 hydroxyproline or 5 hydroxylysine. Now types, collagen subtypes, there are 4 subtypes of collagen, collagen 1, 2, 3, 4. Collagen 1 is present in bones, ligaments, tendons. Collagen 2 is mostly present in cartilage. Collagen 3 is in skin, and collagen 4 is in basement membrane. Now let's talk about synthesis of collagen. Now we know that nucleus encodes DNA, and in DNA after transcription messenger RNA will be formed, and messenger RNA will be coding for collagen. So collagen after translation it will form the protein, and this protein, we know that rough endoplasmic reticulum is site of protein synthesis, right? So this messenger RNA will go to ribosome, and ribosome will translate it in a product which is a protein, and this protein will enter the lumen of rough endoplasmic reticulum, and after modifications it will go to Golgi apparatus, and then it will be sorted and packaged in transport vesicles, and this transported vesicle then will fuse to plasma membrane, and then this product will be released or excreted out of the cell and it will reach extracellular matrix, right? After translation, messenger RNA, the product which messenger RNA will be forming is called pre-pro collagen, and the sequence is glycine XY, right? X is proline, Y is lysine. Later on, hydroxyproline or hydroxylysine will come to this place. So hydroxyproline or hydroxylysine, this is not the essential amino acid, okay? After some modifications, after the addition of hydroxyl group, hydroxyproline or hydroxylysine will be formed, and this process will take place in our body, right? So what happens in hydroxylation, OH group will be added, and this is post translational modification which is happening in endoplasmic reticulum, right? Enzyme used for the conversion are prolyl hydroxylase in case of hydroxyproline and lysyl hydroxylase in case of hydroxylysine. And this hydroxylation cannot occur without vitamin C. Ascorbate or vitamin C is essential for hydroxylation of proline and lysine residue. And if there is deficiency of vitamin C, then there is a medical condition which is called scurvy. Scurvy is bleeding gums because collagen is not synthesized properly, so blood vessels get weak and bleeding gums will be there. So this is due to the deficiency of vitamin C. After that, glycosylation will be there. Galactose and glucose are added to some hydroxylysines, okay? So glycosylation is addition of sugar, and the linkage is specific in case of collagen, and this is O-glycosidic linkage. I'll talk about O-glycosidic linkage and N-glycosidic linkage separately in my next video. And the enzymes involved will be galactosyl and glucosyl transferase. Then after hydroxylation and glycosylation, assembly happens. Pro-alpha chains, as I've told you that, let me take separate page for this. As I've told you that this is like, messenger RNA, okay? So messenger RNA after translation forms pre-pro collagen, whose sequence is glycine X and Y, where X is proline mostly and Y is lysine, right? So after that, this pre-pro collagen is two types of peptide chains, 2 alpha 1 and 1 will be alpha 2, right? These will be having registration peptides and a signal peptide. Registration peptides, what are they? They are basically the signal which tell the protein that they are ready to leave the cell, okay? And with the help of signal peptide, this protein will, like this is ribosome, this is endoplasmic reticulum. So this signal peptide will be somewhere here. So it will enter the lumen of the endoplasmic reticulum, and after its entry into the lumen of endoplasmic reticulum, this signal peptide gets cleaved off, right? So after the cleavage of signal peptide, this product is pro-collagen, pro-alpha chains, right? So these pro-alpha chains undergo hydroxylation and glycosylation lately. So after that, the product will be pro-collagen. Okay, pro-collagen will go to Golgi apparatus where it is packaged. It will go to Golgi apparatus where it will be packaged and excreted by exocytosis. Right? I hope I've made this thing clear to you. So after hydroxylation and glycosylation, pro-alpha chains will form pro-collagen, and this is precursor of collagen. Pro-collagen contains extension peptides at its N and C terminus, and these extension peptides, these are absent in collagen. So उससे पहले कुछ process होगा जिसमें ये extension peptides will be cleaved off. This will be excreted out of the cell through Golgi apparatus, and once it is in the extracellular matrix, this is N terminus, this is C terminus, and this is pro-collagen, right? Pro-collagen peptidase will work here and they will chop off these N and C terminus, and the product will be called tropocollagen, right? So the first step when the protein goes outside the cell will be the cleavage of ends and the formation of tropocollagen. After that, reassemblage of tropocollagen will take place. This formation of collagen fibril happens by association of tropocollagens with each other. So then later on, formation of cross-links. As we can see that cross-linking is going on there. Aldol cross-links. Covalent cross-linking by lysyl oxidase, and this lysyl oxidase is copper-dependent enzyme. And this lysyl oxidase links hydroxylysine and lysine residues. And कहीं कहीं पर histidine बहुत कम, लेकिन histidine का भी cross-linking होता है. So after these aldol cross-links, final product which is formed is called collagen. So collagen is the single most abundant protein in the human body, and the fundamental unit of collagen is tropocollagen. It is a triple stranded helical structure, and this is not the alpha helix. The amino acid sequence in alpha chain is glycine XY, where X is proline and Y is hydroxyproline hydroxylysine. And hydroxyproline and hydroxylysine, these are not the essential amino acids. These are formed after hydroxylation. This hydroxylation is important in maintaining the stability of the triple helix by forming hydrogen bonds between component chains. Talk about some of the medical conditions which happens during the mutation or because of the mutation in collagen. The first one is osteogenesis imperfecta, and already I have talked about scurvy. So this is osteogenesis imperfecta, which is an autosomal disorder, and this happens because of the mutation in type 1 collagen, right? And the result is brittle bones, blue sclera, and hearing loss is there. Second is mutation in collagen type 2, dwarfism and skeletal deformities because of mutation in cartilage. And there is Ehlers-Danlos syndrome, which happens because of the mutation in type 1 and type 3. And in this, weak blood vessels, highly extensible skin, and hypermobile joints because they are prone to frequent dislocations, right? And then Alport syndrome, which happens because of the mutation in type 4 collagen, and eye abnormalities, hearing loss is we can see there, and kidney से related क्या होगा? Basement membrane deteriorates, so proteins and RBC enter the urine. Okay, so this is all about collagen, its types and synthesis, and some of the medical conditions associated with it.